All right, so welcome everybody. Today we're gonna have a journal club on a paper titled Three-Dimensional Fibrotic Extracellular Matrix Directs Microenvironment Fiber Remodeling by Fibroblasts. And this will be presented by Mehmet, who will introduce himself. And after that, we're going to have a lively discussion by a panel of experts, as well as by audience. So a panel of experts are made up of Prof. Amanda Tatler and Dan Weiss. Prof. Amanda Tatler is from Nottingham, and Prof. Dan is from, I'm trying to find my notes here, which is missed, unfortunately not opening, sorry. Dan, if you could unmute yourself, and I'm gonna ask both all of us to introduce ourselves because my nose is actually crashing here. So we're just gonna have, the way I'm gonna do it now, because my nose is crashing, is I'm just gonna let us all introduce ourselves, and then I'm gonna hand it off to Mehmet for him to start with the presentation. I guess that'll be the best way because my nose has crashed here. So I'll hand it off to Amanda for you to introduce yourself, and then Dan will continue the introduction, and then Mehmet can begin.
Sure, thanks, Emmanuel. I'm Amanda Tatler. I'm an associate professor at the University of Nottingham, and I'll be acting as one of the expert panelists today.
Thank you.
Very nice. I'm Dan Weiss. I'm a professor of medicine and bioengineering at the University of Vermont, and we have a long history of interest in lung regenerative engineering and medicine, and in particularly the effect of the environment on lung cell behaviors. And I'm sorry to say I'm not an RSF member, but rather RCMB, co-RCMB. So anyway, I'm delighted to be doing this, and thank you for asking.
We are very happy to have you here, Dan. And I'm actually happy that I did it this way, because okay. All right, so and I'm Emmanuel. I'm an assistant professor here at UBC, University of British Columbia, also very interested in models to study lung diseases. And so very happy to moderate this. So I'll hand it over to Mehmet. Mehmet will give us a short introduction of himself, will present the paper to us in about 30 minutes. After that, expert panelists will discuss the paper, and we'll have time to ask him some questions, and then the audience can then join in and ask questions. And so I'll hand it over to Mehmet.
Thanks, Emmanuel. Yes, my name is Matt, and I also known as Mehmet. That's my birth name. I respond to both, and I have done my PhD on three-dimensional models and how we can use these models to examine and understand how extracellular matrix interferes with the disease progression or contributes to that. And today I am very excited to show you some of our results that we published last year in Acta Biomaterialia. I would like to start by sharing my screen. I hope that everyone is able to see the screen.
Yep, we can see it.
Perfect, thank you. All right, so as I said, I'm very excited to be here, and this research was performed at the University Medical Center in Groningen, Netherlands, at the Department of Pathology and Medical Biology. Before we start, I would like to give a brief conflict of interest declaration that this study was financed by an unrestricted research fund through Boehringer Ingelheim Pharma Company. So our main topic today will be in the context of idiopathic pulmonary fibrosis, or IPF in short. IPF is a deadly disease that starts at the distal end of the lung tissue and results in the shortening of breath of the patients. But I would like to talk to you through what happens. In a healthy scenario, normally we have beautiful alveolar structure where the interstitium is thin and letting the exchange of oxygen and carbon dioxide between the alveolus and blood vessel nicely. Once the IPF manifests, once it progresses, we are seeing excessive extracellular matrix deposition in the form of fibrosis, resulting in the dilation of bronchi and causing alveolar remodeling. This, in the interstitium, shows itself as thickening of the interstitium and the deposition of the scar tissue in this now thick interstitium. This eventually results in the hindered gas exchange and causes the patients to have shortness of breath, to say the least. More on the details of the disease, we know that 5-year average survival of this disease, of these patients, is around 20%. And not only that, but we are lacking curative treatments. The only final cure for these patients is a lung transplant. Not only that, we don't have enough treatment options, we are also lacking representative animal models to be able to understand how this disease starts, how this disease progresses. This puts us in a difficult scenario because we still need to understand and improve our understanding on how this disease progresses. I have mentioned that there is a deposition of extracellular matrix, or ECM, in the form of fibrosis. This fibrotic ECM is known to be quite different, and we can categorize these changes in 3 major components. That will be ECM composition, ECM organization, and ECM stiffness or biomechanics. I would like to dive into each one of them in the upcoming slides. Starting by the ECM composition, you can see the comparison of schematics for healthy and fibrotic lung extracellular matrix environments. One of the first things that I would like you to notice is how crowded everything it appears in fibrotic lungs, which is true, because we see an increased production and deposition of extracellular matrix proteins in fibrosis. So things are much more crowded. The protein amounts of collagen 1, collagen 3, elastin are known to be increased in the fibrotic microenvironment, and this gives the increased amount of binding sites to the cells, which are also known to be increased in sites. So we are talking about a more crowded environment, more things to attach around for cells, and more domains for fiber cross-linkings to happen. These cross-linkings are shown in these blue star icons, and they have implications for ECM organization changes, which brings us to our next slide. On the slide, you can see 2 images on panel A, and these are obtained through an advanced microscopy method called second harmonics generation, and it shows us how the collagen is organized. In cyan color, it is showing us the organized collagen, and in yellow, it shows the disorganized or young collagen, immature collagen. What you can see already is when you compare non-IPF to IPF from left to right, you see a big discrepancy towards the organized collagen. This organized collagen ratio, when we take them, we see a big increase towards the organized collagen level, and this tells us that the fibers are much more tightly linked to each other, creating this tight network of proteins in which cells are attached, maybe almost like a prison-like. This tightly weaved network results also changes in the stiffness. On the slide, you can see in panel A, the comparison of normal and IPF lungs in terms of their stiffnesses. I would like you to appreciate the logarithmic scale here and see that there's a 10-fold average stiffness difference between normal and fibrotic lungs, which means 10 times harder. And once we remove the cells and all cellular contents through decellularization, we see that the lung extracellular matrix proteins as a scaffold is still holding this trend and approximately still 8 to 9 times harder, stiffer than the normal lungs. All of these tell us that there is a big difference in the fibrotic ECM compared to the controls. But this is not where the story ends. This is only where it starts. Because we know that lung extracellular matrix is also instructive. In 2014, Parker and et al. Have done these beautiful experiments in which they have put control fibroblasts and IPF fibroblasts on top of both IPF and control environment derived from cells. And they have checked the RNA production amounts. Once they compared the cell origin, they did not see a significant difference, so the values were quite comparable. However, when they checked the influence of ECM type, that would be the ECM coming from IPF or control, they have seen a big increase resulting from the source of ECM that these cells were seeded on. Later in 2018, Phil et al. Checked this in terms of ECM cross-linking, especially in the context of LOXL-like 3 protein, which is an enzyme that creates and cross-links the ECM further, therefore changing the organization. They have seen that the matrix effect, when compared to IPF to control, they have seen 4 to 5-fold change in the amount of enzyme produced, only coming from the fibrotic matrix itself. This told us that lung ECM triggered further disease phenotype. But this wasn't the only key factor, actually, because this created a paradigm shift in how we are looking at the disease and our understanding of how the disease perpetuates. Because now we started thinking about how ECM works and how ECM is influencing this process. All of these studies were pioneered in 2D environment. In these early studies, we have a polarity because the cells are seated on top of a plastic environment, which brings us in terms of high stiffness. And they are also adhering to the environment only through one direction. On the other hand, in fibrosis, or in our bodies, the cells are experiencing a 3-dimensional environment where there is no force polarity. The stiffness can be different in all 3 dimensions in which the cells can attach to. Because of this, even though these pioneering studies paved the way for our understanding, we need to think about the matrix in 3D. To summarize the problem, we know that the ECM changes are both result and the reason of the disease progression. But our understanding of how ECM remodeling fits into this cycle is not yet established. To be able to establish this, we need alternative in vitro models. That brings us to the overall hypothesis that we had in our study. And we hypothesized that the 3-dimensional fibrotic microenvironment instructs the cells towards a profibrotic activation. To investigate this, we have used a novel methodology called lung extracellular matrix-derived hydrogels. To obtain these hydrogels, you start with native lung tissue that can be both from control and IPF lungs, which was the case for our study, and you remove all cells and cellular contents through steps of washing with chemicals and detergents, resulting in decellularized lungs. These cell-free lung tissue contains all the extracellular matrix proteins in their correct proportions in IPF or control, and you can grind this to create this fine powder. The powder of these lung tissues can then be digested using pepsin, and this solubilized lung ECM forms these beautiful hydrogels, which we can use for 3-dimensional cell culture. And these hydrogels of either control or IPF lung origin was used throughout the study, and it will be coming back in all the results. We have seeded these hydrogels with fibroblasts coming from control and IPF origin, and we have used empty hydrogels of control and IPF as our baseline measurements to be able to see whether our hydrogels are showing the same trends that we observe in the native lung tissue. Once we had these cell-seeded hydrogels, we have cultured these for 7 and 14 days period, and then we have characterized them for the following. We have measured the collagen staining area and collagen staining intensity in terms of quantifying the ECM composition. We have checked the collagen fiber organization and characterized them to be able to see how ECM organization changed over these 7 and 14-day culture period. And last but not least, we have investigated the stiffness and stress relaxation behavior of these hydrogels to be able to see the ECM stiffness or biomechanics. Before I delve to the details of the results, I would like to talk to you how we have reached these conclusions and how our analysis strategy was. For any individual measurement, from these 6 groups, we have obtained 6 different data. All of these graphs that I will be showing in the subsequent slides are mock data, but in essence, once we have our individual measurements, we have performed a mixed model analysis that resulted in estimated means and their confidence intervals, which means that the y-axis is not the actual representation of the actual measurement anymore, but the data trends continue holding. From these estimated means and confidence intervals, we first established our baseline differences. This means how empty IPF hydrogels differed from control empty hydrogels for any given parameter. For all of the subsequent graphs in the results section that I will be showing you today, this baseline difference will be shown as the dotted lines in the graphs. When it comes to the responses of the cells, we wanted to see how cells change the environment further over the existing values. For this, for example, on the left side, you can see how control fibroblasts in IPF hydrogels elevated this particular parameter readout over the existing values. And these values were regarded as their responses towards IPF hydrogels. We have performed the same calculation for the control hydrogels, and we recorded these as how they behaved in these respective environments. Then we took this one step further, and we have compared these 2 responses. And these responses are going to be shown to you today in the form of light blue triangle in the case of control fibroblasts. And every graph will tell us how these fibroblasts responded in IPM environment compared to control environment over their respective baseline values. Parallel to this, we have done the same treatment to IPF fibroblasts, and in all graphs, they will be shown as dark blue rectangles, and this will tell us how these fibroblasts responded in different environments. To sum all of these up, in the results section, you will see graphs looking like this. And the first thing that I would like you to notice is the baseline differences in empty gels. This tells us what was the case of IPF hydrogels compared to control hydrogels. And the next thing that we would like to look at actually is the y-axis, spotting the 0, because anything above 0 will mean to us that the groups responded higher in IPF gels, and similarly, anything below 0 will tell us that these groups responded higher in control gels. For example, here we can already say that IPF fibroblasts responded strongly in control gels, and control fibroblasts responded strongly in IPF gels. To be able to see how different they were over the existing baseline differences, we have performed a statistical analysis, and the results of these analysis in the form of p-values will be shown below the columns. This will tell respectively the control fibroblast responses compared to baseline and IPF fibroblast responses compared to the baseline. Last but not least, we have compared these two fibroblasts with each other, and this comparison will be shown on top of the graphs for any result to be able to see how control and IPF fibroblasts differ from each other in terms of their responses in these two different microenvironments. Moving on to the results, the first result would be about the ECM composition, which we assessed by using picoC's red staining towards collagens. When we looked at the day 7 results, we have seen that the existing differences between the empty hydrogels was towards a fibrotic domain, so it was above 0, and the cells did not additionally add any collagens detectable. This resulted in non-significant differences over the baseline and among the groups. Once we checked it for day 14, we have seen that all of our measurements were in the fibrotic domain, as seen by the purple background in the graph. However, these differences still were not beyond the existing baseline, and they were not different in different groups. About the interpretation of these results, I will come back at the discussion part. But for now, what we can conclude is that fibrotic microenvironment did not cause any detectable collagen change in both control and IPF lung fibroblasts. Moving on to the ECM organization, the first parameter that I will be talking now is called high density matrix, and these areas are defined by these highly entangled clusters of collagen fibers. Therefore, a higher area percentage means that there is more of these entangled proteins, and lower area means that there is less. Once we quantify these percentage areas, we have seen that our hydrogels, to begin with, when they are empty, there wasn't a big difference. It was slightly higher in control hydrogels, and our cells responded differently. You can already appreciate that collagen fibroblasts increase the high density matrix in IPF gels well over the baseline difference, while IPF fibroblasts did not result a noticeable increase or decrease. Their differences also were significantly different, and we can say that there's already a cell and ECM combinational effect in the regulation of higher dense part of the matrix. Once we moved to day 14, however, we saw that the trend continued on the opposite direction. Both of these cell groups responded higher in control gels, which means that they decrease the high density matrix in fibrotic microenvironment. Their responses were significantly different than existing baseline, and IPF fibroblasts were influenced by the fibrotic microenvironment stronger than the control microenvironment, as shown with the significance. Therefore, we can conclude that both of the fibroblast types were strongly influenced by the fibrotic microenvironment to decrease the percentage high density matrix. The next category of the ECM organization that I would like to talk now is the fiber alignment percentage. This, as you can see from the example fluorescence micrographs, it tells about the percentage of fibers that has similar or different fiber alignments. Here on the left side, you can see that the fibers are randomly aligned. On the right side, you can see that these fibers have a similar alignment values. So our parameter measured how much of the percentage fibers have the similar alignments. Once we check the day 7 results, we have seen that IPF hydrogels, when they are empty, were slightly more aligned compared to control hydrogels, which is shown by the baseline difference. And control fibroblasts did not change the alignment over the existing baseline. However, IPF fibroblasts strongly responded in IPF gels and created a more aligned microenvironment for themselves. As a result, this resulted in difference between the control and IPF fibroblasts. When we checked day 14, we have seen that this trend continued and IPF fibroblasts kept and further drove the alignment towards a higher alignment, while control fibroblasts did not change the alignment values. Therefore, the summary would be that IPF fibroblasts alone were instructed by the fibrotic microenvironment to drive the alignment to a higher values. Moving on to the last category of readouts, the ECM stiffness. When we measured it for day 7 samples, we have seen the following results. You can already appreciate the fact that everything happens in the IPF domain, shown by the pink background, and even the baseline difference was well over 0, which tells us our IPF hydrogels were much more stiffer compared to control hydrogels. Once seated with both type of fibroblasts separately, these fibroblasts drove the stiffness further and resulted in a significant increase well over the baseline. By day 7, we saw that these fibroblasts responded comparable values. However, on day 14, we have seen that control fibroblasts responded much stronger and drove the stiffness values much higher in the IPF gels compared to baseline differences. This was not the case in IPF fibroblasts, and because of these, we can say that they responded differently in fibrotic microenvironment, which brings us to the conclusion of that fibrotic microenvironment instructed the control lung fibroblasts to increase the stiffness, while fibrotic lung fibroblasts remain the same. Parallel to the stiffness values, we can also check how the applied stress is relaxed. And when we looked at the day 7 values, we have seen that even though everything was in the fibrotic domain, which means that the fibrotic gels relaxed off of the stress much slower rates, by day 7, all of the values were comparable, and the cells did not modify the microenvironment regarding this capacity. However, on day 14, both of the cells increased the duration that it takes for these gels to relax the stress, and in the case of control fibroblasts, they responded stronger over the baseline in the fibrotic microenvironment, which brings us to the conclusion of that the fibrotic microenvironment caused these control lung fibroblasts to decrease the stress relaxation and increase the duration, while fibrotic lung fibroblasts did not result in any change. To summarize all the changes that I have talked so far, we have investigated the changes instructed by the fibrotic microenvironment in 3 different categories, which were ECM composition, ECM organization, and ECM stiffness, to mimic how these changes occur in the lungs of patients with IPF. We have checked the collagen amount in terms of collagen ECM composition, and we did not observe any detectable differences. We think that this is because the primary material of these hydrogels come from the lung matrix is collagen. And since there is such a predominantly high amount of collagen in the hydrogels to begin with, any value that was added or deducted from this collagen by the cells was not detected by our method. And to be able to detect this, we need further, more sophisticated ways of checking how the collagen amount changed. In terms of ECM organization, we have checked the high density matrix and alignment, and we have seen that both cell types decrease the high density matrix while IPF fibroblasts increase the alignment percentage. And in terms of ECM stiffness and biomechanics, we have seen that control lung fibroblasts increase the stiffness and decrease the stress relaxation, while IPF lung fibroblasts did not change these parameters significantly. Here we can see the pattern forming already, and we can hypothesize that the IPF lung fibroblasts coming from a fibrotic matrix, they already have the fibrotic microenvironment around them, and they don't need further changes to make it more comfortable for them, so to speak. While controlled lung fibroblasts being healthy, they are much more naive and much more malleable towards the instructions coming from the fibrotic microenvironment, and they drive the environment towards a more fibrotic microenvironment, especially in the increased stiffness and decreased stress relaxation values. Therefore, we can say that our model was successfully replicating the changes happening in terms of ECM organization and ECM stiffness. To conclude all of these, we can say that our fibroblast-seeded ECM-derived hydrogel model was able to recapitulate the fibrotic lung microenvironment in a 3D capacity. It demonstrated the temporal and differential responses of these two fibroblast types in both control and fibrotic microenvironments. And last but not least, these hydrogels highlight the ECM organization and stiffness as potential therapeutic targets. And this is quite important because, as I said in the beginning, we are lacking alternative treatment methods and cures towards IPF or any fibrotic disease. And knowing that ECM organization and stiffness can be potential targets gives us hope towards checking further avenues in terms of therapeutic targeting. And not only for IPF, but also other fibrotic diseases, such as kidney fibrosis, skin fibrosis, or liver fibrosis. With this, I have reached to the end of my presentation. I would like to start thinking to patients and non-disease control donors, their families, and all clinicians, surgeons, and other hospital personnel who made this research possible. And after that, I have the privilege of working with a talented group of researchers. I would like to thank them as well. And I would like to thank you for your attention. And I'm happy to answer any question. Thank you.
Thank you very much, Matt. And I always mess up because I always call you Mehmet. So
no worries.
Thank you very much, Matt. So at this point, we're going to invite our expert panelists to give their take on your presentation and your paper. And this will take the form of anything they see fit. They can give comments by putting your work into their perspective in general. And so we're going to give both Prof. Dan and Prof. Amanda 5 minutes each to give their take on your paper. And this, apart from giving their take, they could also then ask you questions. After this, the audience can then join in to also ask you questions. Dan and Amanda, do you think this is enough time for your comments?
Not even close.
Okay, so I'll hand it off to you. Let's start with Dan.
Well, no, Amanda, please, if you'd like, go ahead. I do have a lot of comments. So, but one thing I want you to think about, Matt, is chicken and egg phenomenon here, right? And we'll come back to that. But please, Amanda.
Yeah, a great presentation of your paper, Matt. I really enjoyed it. And I think these 3D models where you've taken the matrix of a patient to build your hydrogel has to be much more relevant than a matrigel system or some other GELMA-based system where we're adding in what we think are the right components, right? So I want to applaud you and the group for that, because I think it is a huge advance in the field of fibrosis research. You gave a really nice summary at the end, and I'm just wondering if I could draw you out on what you kind of hinted that maybe the IPF fibroblasts don't really respond in the same way as the control ones because they're remembering the environment that they were in. And I wonder if I could draw you on what you think that might be. How Well, a few technical questions, I guess, is that how were the cells cultured? How long is it since they've been out of the fibrotic environment within the lung? What passage, etc. And if they are remembering that they used to be in a fibrotic environment, and then you put them in a fibrotic hydrogel, and therefore they don't respond because they're used to that environment, how are they doing that?
Oh, thank you for your kind words, Amanda. And I'm happy to brainstorm on this because this is an ongoing topic of discussion, how we culture these cells, right? You're right, they have been out of the lung tissue. So they are grown through explant culture where we have a small lung tissue and we expand these fibroblasts. When they were used in the experiments, both control and IPF lung fibroblasts were at passages 4 to 6. We kept this comparable for all the donors, and unfortunately they are grown on plastic, and there is a certain imprinting coming from that. And until we find alternative methods to expand the numbers of fibroblasts on anything but tissue culture plastic, unfortunately this is going to be one of the major limitations where we try to see the effect of either chemical or biochemical composition of the extracellular matrix.
Yes, it's a huge shame, isn't it? I was talking to a colleague and collaborator recently about, with us not really having a really good fibroblast marker, we can't even, they always have to come through this explant culture, right? We can't just single cell sort the lungs and take them straight from the lungs into a hydrogel, which is a real pain for fibroblasts.
That is definitely true.
So you think that the being in the fibrotic lung is just changing them in some way? What are you following that up? How might they have remembered that they used to be in a fibrotic lung?
I think it starts by actually. So now we are talking about, of course, as if they are human, but that's on me. I started by saying that they remember, but I think they are being programmed, so they are receiving all these instructions from the stiff microenvironment that triggers certain mechanotransduction events in the cells, and it creates cascades of synthesizing more collagen, for example. This is one way of doing for them remembering that they were in the fibrotic microenvironment, because they have received instructions to produce more collagen to begin with, or cross-link it further. So because they were in the process of making these more cross-linking enzymes, more collagen molecules, and deposit it to the extracellular space, this is how they remember that they should be acting further fibrotic. But because in the fibrotic hydrogels, there has been enough stiffness and there has been enough collagen molecules, I think this stopped them from acting on this further. They didn't need to adjust the microenvironment further because it was already fibrotic enough. They were saturated.
Okay. And have you looked at this is a really nice body of work. Have you looked at any signaling changes in the two cell types in the different hydrogels?
We have attempted, I could say. Checking RNA level measurements in these hydrogels have been proven quite difficult, unfortunately, because lung ECM hydrogels have this extraordinary capacity to hold any RNA that is around to them. Therefore, isolating RNA from these hydrogels was not possible. After 40 or 50 different methodology trials, I unfortunately gave up. I'm happy to take suggestions on that. So in RNA level, we couldn't check. In terms of soluble factors, again, ECM is known to be the reservoir of growth factors. So some of the growth factors we attempted to check were not showing any signals to us, again, because of the binding capacity of matrix. So characterizing these hydrogel environments for soluble RNA and protein levels was quite difficult. But that will be quite interesting to check how these cells are communicating in paracrine factors.
Mm-hmm. Okay. And then another thing that struck me, I think, so at the moment you're creating fibrotic and control hydrogels from completely different donors. Have you tried? IPF is really heterogeneous. in one patient, you might have a very fibrotic bit and then a bit that looks very histologically normal. Have you tried doing the same patient, but a fibrotic area and a control area?
You're saying actually a very true part for the disease progression is quite heterogeneous, and because of that, we try to normalize it by grouping the donors. So for control and IPF donors, we created the mixtures to balance those differences. For within patient differences, we did not do that. It sounds actually a quite cool setup for an experiment. Unfortunately, the amount of samples per patient that we are obtaining was quite limited to milligrams, and that did not allow us to do donor match setups. Unfortunately, I would love to do that.
Okay.
It's technically challenging, right? Because I think you're going to need a scan associated with the patient as well to make sure that you're taking what you think is the really fibrotic bit. But I think that would be really cool because you can completely taken out any donor to donor variation. Then you are anyway. Dan, you're talking at the bit.
Yeah, exactly.
So, no, those are really good points, Amanda, and some of the ones I was hoping to make. So, Matt, also congratulations, both on a great presentation, but also a solid body of work over a number of years and the like. But I want to take up on a couple things that Amanda has articulated. So the first is the fact that you're pooling lungs. And it is, of course, of interest to look at individual lungs and try and correlate behavior of your product gel with what you know about the characteristic of the patient lung. But that's a huge amount of work, of course. And so I can appreciate the rationale for pooling. But I was a bit confused by the comment and on this slide, actually, that the collagen content on average was similar between the control and the IPF lungs, right? And so that's a surprise, isn't it? Because you really would expect higher amounts of collagen. So part of this is going to get into some nitpicking about your methodology, but how did you measure the collagen? Was it all by Sirius Red and then visual and quantitative visual analyses of, for example, the Sirius Red staining? And then what exactly does Sirius Red stain for? Is it all collagens or is it type 1 and type 3? Because I think you know that some other collagens, like type 6 collagen, surprisingly is really quite preponderant in lung, more so than people expected. And so are there differences, and what does that mean functionally? Take that a step further. If you go to your histology slides, where you're showing the densities, if you will, I think it's slide 21, give or take.
Would this be this?
Yeah, so this is good, right? Perhaps I missed it, but how did you define a low density versus a high density, and how did you create those? One of the issues with working with hydrogels, as you well know, is that the various physical and organizational factors interact. So could it simply be that there's more protein, total protein, in the high density versus the low density, and that plays a role in the cell behaviors, if you will, and the subsequent remodeling or not of the hydrogel. So why don't you tackle those, and we can get further into other things.
Yes, I'm happy to answer all of them. Thank you for the nice compliments. So regarding the collagen detection, we did indeed a Picro-Series red staining on the sections of these hydrogels, both cell-seeded and empty. And then we have digitally captured the image of these sections and digitally quantified, and we categorized them in weak signaling, medium signaling, and intense signaling. And we did comparing all of them on an automated manner. So it was done in a digital quantification. And you're right, we did not find any difference, and we were expecting to find much more collagen in IPF environments, because that is what we know from the IPF lungs coming from the patient material. How I can explain is that These hydrogels are starting with similar material. To go into the details, we start by digesting 20 milligrams of control or IPF lung matrix in powder form in per milliliter enzyme. So we know that most of the lung proteins are collagens. Therefore, you will have 90% collagen, 20 milligrams starting material. That results in a very high abundance of collagens. And I think the changes that the cells might have introduced got lost in this abundance of collagens because there's so much that the picoseries red stain that stains all collagens, as far as I know, it just overpowers every small change that might have been there, either in the form of decrease or increase. I mean, that would be cool to, for example, provide labeled amino acids to see how the new deposited collagen is incorporated in the existing environment, or we can fluorescence label that. But we didn't expect to see the lack of changes.
Yeah. So just to elaborate on that, yes, the majority of the total extracellular matrix proteome is collagen of one type or the other, right? And so if you didn't see a difference in your base material, right? Had you done, and I think you have, for example, mass spectrometry, and have a better sense of the portfolio of proteins, and is there a difference that you could point a finger to? Because yes to collagens, but all these other proteins have potential significant functional roles. And you have a great model system to potentially tease out what is necessary and or sufficient to drive fibrotic cell behaviors in a control cell or not in a fibrotic cell, which is what you seem to have found. So what do you think about that?
Yeah, in terms of the other proteins other than collagen, I think we need to start looking into them for sure. Indeed, proteomic analysis of these hydrogels in a different setup is an ongoing project. But at the point of finalizing this study and writing the manuscript, a mass spec wasn't in the plan because, as I said, we expected that, we are going to see lots of collagen. We also checked glucosaminoglycans, which are known to be different in the IPF, and we didn't see any differences in our samples with the method we used. So these were, of course, methodological choices that we had to make at the time. Now with this information, we can do a more educated guesses for the next method that we want to apply. And yeah, mass spec and checking further details of the proteomics is definitely something that we are also interested in.
So, Matt, make no mistake, this is all very positive. I'm just
yeah, I know.
No, help you think about, because again, it's still no difference in collagen in your baseline samples. Just again, it makes it more challenging to understand the results. But I also want to go further into the two photomicrographs here, right? So the low versus high density. So can you explain better what those actually are? And as a corollary question, right? If you look at for example, the low and the pico series red staining, there are areas of actually a significant portion of your gel seems to have no collagen in it, based on that metric. And so one thing that would be of really interest, where do the cells lodge? If you had, for example, histologic sections stained just for nuclei or what have you, Amanda's point about, how do you identify fibroblasts, of course, right? But where are the cells distributed, and is that different between the low versus high? Is that difference between your control versus your IPF gels? And do you think that contributes to, again, the endpoints that you did measure?
Yes, I'm happy to talk about these two images. So these were fluorescence imaging on the picoseries red, because it also emits a fluorescence wavelength when you just looked under the fluorescence microscope. So we didn't do second harmonics on these particular samples. And how it was measured is that the software that we have used called Twombly, and I'm happy to share the details later if it's required, creates these tracks basically every existing fiber. And then it starts checking where these particular entanglement of fibers might occur.
Yep.
It measures these, or it basically envelops these areas and then removes them from the equation. And then it checks every fiber area and calculates these areas based on the amount of free floating fibers. So yes, that's a very valid observation that there are lots of gaps. And I think that was coming back in our results as percentage high density matrix in the controls even being also emptier. So this is, but the gaps are not the lack of high density matrix. The lack of these kind of, so to speak, cityscapes in the night type of very dense populations of matrix fibers is the high or low high density matrix.
So what you're saying, if I understand correctly, is that if you showed us images of the different intensity emission, if you will, from the picoseries red, that the black areas, for example, on the low density photomicrograph wouldn't be black. There'd actually be staining indicating that there is collagen in those apparently empty spaces. Is that?
No, not exactly. If it sounds like that, then I said it wrong. Maybe in the below in the different Z stacks there might be collagens because these hydrogels were 3 millimeters thick in the end.
Yes,
So there is definitely high collagen above and below. But this particular example, yes, there is no collagen in this exact Z stack in this area. But this gap was not regarded as low high density matrix. The low part comes.
I'm going to have to jump in here and throw it to the audience also because we are almost fine. We have about 5 minutes. So I'm going to have to ask the audience, if you have questions, you can please jump in and ask your question. You can also write your question in the chat so that we can, we can also take your questions also. I think this has evolved into a discussion between the panelists, and which was expected. But if there is any burning question by the audience, we would also take the questions too. Thank you.
Hi, Mehmet. I'm Niyati. Nice to meet you. Nice to see you.
Hi, Niyati.
Yeah, I went through your work too, exceptional, and I'm really happy to, attend your talk today. So I have one question which is connected to both the ECM organization and the ECM stiffness. With respect to your stress relaxation measurements, do you think the ECM, like what ECM remodeling activities do you think are different between the control fibroblasts in the fibrotic environment at day 7 as opposed to the IPF fibroblasts, which possibly lead to the decrease in stress relaxation that you see? Do you think they are more related to like the collagen cross-linking or collagen alignment? And as Dan mentioned, do you think there's like a certain collagen isoform that is probably playing a role there? And based on that, do you think the observed changes in stress relaxation can be linked to, the fibroblast behavior? Basically, can this increased alignment that you see lead to altered stress dissipation that you see in IPF fibroblasts, which thereby lead to like reduced stress relaxation? So can you comment on that part a little?
Definitely happy to comment on that. So I think how the stress relaxation is decreasing in the control fibroblast seeded IPF gels is through also increased stiffness. So we know that these two phenomenon happen most of the time simultaneously, and it is a puzzle to solve actually, to modulate them without the other one being interfered. So I think the lack of alignment and increased stiffness is creating this tense environment. So these are the ECM remodeling activities coming from the cells that results in the decreased stress relaxation. But what you are asking and what Dan pointed out also about particular collagens or fibrillin or decorin, these are proteins that are very key to ECM organization and function. function. So we did not have enough time and capacity to check all of these to be able to say, are these also pointing out important differences in alignment and relaxation. That would be very cool to check, for example, decorin values, other than something with staining, of course.
And I think also with the introduction that you gave, I think the different types and distribution of these glycans or proteoglycans that you're seeing could alter, the fibroblast-driven collagen fiber alignment possibly that you mentioned you will want to look at in the future. So thank you, Mehmet.
Yeah, thank you.
Congratulations again. Thank you.
Thank you.
So we have just about a minute, but there's a comment in the chat, a question asking about whether you've added other cell types to the gels, like immune cells. And Jeanette has also commented to answer already that you are now doing experiments with monocytes.
Yes,
as to be
indeed.
expected.
That is creating the microenvironments more in the sense of fibrotic and controlled microenvironments. Those are the next steps that we are taking.
Well, and as you well know, we're all working together with Jeanette on alveolar cells in these gels.
Yes, I think epithelial cells would be very cool to add. I just saw that Amanda added that as well to the chat. Yeah, that's definitely true. These hydrogels, I mean, the 3D microenvironment is of interest to all cell types. But the problem is, once you have more cells, then you need to cater towards their needs. This comes with media complexions. This comes with stiffness problems. How do you expand them? Do you have them donor matched, or how can you culture all these cell groups at the same time? I think as the field, we need to come up with either better ways of overcoming these challenges or lowered thresholds of what we expect so that we can increase our knowledge one step at a time.
So Matt, in the last minute, if I may, Emmanuel, let's get back to the chicken and egg. So what do you think?
Yeah, that's definitely a chicken and egg situation here. I would say that
you have to pick one. I'm sorry.
I would have to pick one. So what's your question then? Which one is chicken and which one is the egg for ECM and the cells?
Well, which comes first? Exactly. Yeah.
Which comes first? I would say fibrotic matrix comes second. So the cells are becoming fibrotic first, then producing fibrotic matrix, then results in further conversion of the naive cells. This would be my educated guess. But unless we generate more body of information, I do not know for sure. My pick is that cells are first.
So to have more fun with you, right? How would you design that experiment?
You're throwing me at a corner here. I would do a series of experiments in which, for example, the organization and composition and stiffness gradually changes separately, so that you can actually tell where the cells start acting differently. That would be one way of doing it. But don't ask me how. I do not know.
Well, thank you so much. This has been a very lively discussion. And thank you very much, everybody, for joining and for your questions and, contributing to this discussion. Hopefully, this has stimulated some, amazing thoughts, and we'll watch this space and add more to this. Thank you very much for the presentation, Mehmet, and thank you to our two wonderful panelists. Thank you to the ATS supporting staff for organizing this, helping to organize this. And thank you everybody for joining, and happy Easter.
Thank you all.
Thank you all. See you.